Welcome everybody to today's edition of the EV Journal Club. We have a very nice presentation for you today. And we also have a guest co-moderator, Tom Driedonks, who is a postdoctoral fellow in my lab. So I'm going to hand this over to Tom to introduce our speaker. Tom, thanks for moderating today.
Thank you, Ken. It's really an honor that I can moderate the Journal Club today. So today's speaker is Agustin Enciso-Martinez from the University of Twente. He's been working together with Professor Leon ter Stappe at the Department of Medical Cell Biophysics. He does research on extracellular vesicles derived from cancer cells. And today he's going to share with us his most recent publications, which is about label-free identification and characterization of single EVs and lipoproteins. So I read through these papers. It sounds like really cutting-edge technology, so I'd love to hear more about it. So Agustin, can you please get started?
Thank you very much, Tom, and thank you also very much, Ken, for the opportunity of presenting this work. As you mentioned, I will talk today about some of the methods and techniques that we have been using for the detection and characterization of specifically tumor-derived extracellular vesicles, but also lipoproteins. I work doing my PhD a lot in this in the medical cell biophysics, trying to work on these methods for liquid biopsies. So I will present this work that I have done during the last years. So let's start. Well, the title is long, but I will go through it. It's using synchronized Rayleigh and Raman scattering to characterize single extracellular vesicles and lipoproteins. Let me just first go through the outline. So I will first give a brief context on the tumor-derived extracellular vesicles and the liquid biopsies. Then I will move to explain what is Raman spectroscopy, one of the techniques that I have used a lot to characterize the chemical composition of the vesicles. And then I will describe the methods that we have worked on to detect and characterize single particles. The first one is we use scanning electron microscopy, Raman, and atomic force microscopy to characterize the vesicles. And then the second one, we trap the vesicles with the light, and we perform the Raman characterization to look at the chemical composition. We started using first model particles, such as polystyrene beads, silica beads. Then we moved towards vesicles from established cell lines to finally we do work with plasma and particles that are present in the plasma of healthy donors or prostate cancer patients. This was a small pilot study with a small cohort of patients. And then after the conclusion, I will raise some discussion points. So, well, most of the people here are very aware of what are extracellular vesicles and that all the cells in our body are releasing extracellular vesicles. And the release of extracellular vesicles has been a pretty evolutionary conserved process across species, and it has been seen that is one of the purposes is for waste removal, but it can also have a communication purpose. Now, cancer has not been the exception, and cancer cells also release vesicles, which have different kind of biomolecules, and these biomolecules can perhaps reflect the nature or tell us something about the parental cells. For instance, proteins that are in the vesicles that can be also found in the parental cells. So here we can see an opportunity as a biomarker, because if, for instance, the tumor-derived vesicles are released towards the circulation, we could detect them and maybe look at the specific markers that, and then see where is the primary tumor, for instance, where is the tumor located, which tissue. So I have seen many presentations from the EV Club, and they have shown very beautiful images about vesicles and how do they look. We saw in this case, this is just a snapshot of a cell, and the cell is releasing extracellular vesicles, as you can see here, of different sizes. This is taken by electron microscopy, scanning electron microscopy. And there might be some apoptotic bodies, some maybe ectosome, exosomes. We don't know because this is just some imaging, so we cannot confirm biogenesis. But it already shows a very broad size range. And then here on the right, we see a set stack of cells that took up vesicles that are here shown in green. So just to give a little bit context on the metastasis and the role of different actors, such as tumor vesicles. Normally, one of the key actors in the metastasis have been the circulating tumor cells, which can actually enter the circulation and at some point extravasate and establish a secondary tumor and another tissue. But in the literature recently, has also been mentioned and described that cancer cells in a tumor can also release extracellular vesicles, which we call tumor-derived extracellular vesicles. And they can enter the circulation via transcytosis or perhaps by weakening the junctions between the endothelial cells. They can circulate, and at some point they can extravasate and create the proper conditions for a secondary tumor to establish at that site. But hopefully before that happens, we can, for instance, take a blood sample and analyze and detect these tumor-derived vesicles, for instance, to see where the tumors are located, in which tissues are they located. Or we could maybe enumerate the tumor-derived vesicles to say something about the prognostic of the patient. So, or even better, we could use the vesicles and characterize them to select an optimal therapy. So we could use the tumor-derived vesicles as cancer biomarkers. And the goal of the work that I will be presenting was to detect and characterize single tumor-derived vesicles in a liquid biopsy. But there are actually, it is not that easy, and there are many problems involved. And one of the main problems is that in the plasma, there are many other particles that are overlapping in size and density with vesicles. And these are, for instance, lipoprotein particles. And they are a lot more abundant than the vesicles and even more than the tumor-derived vesicles. So if we have, for instance, if these are all the particles in the plasma, and we are trying to aim at to look at rare populations of vesicles, such as the tumor-derived vesicles, then we are aiming at finding our Waldo balloon. And for this, we actually need single particle analysis techniques, because you can imagine that bulk analysis techniques can actually average out the properties of the tumor-derived vesicles, and they can hide these rare populations of vesicles by the ensemble average, for instance. So with this goal in mind, we try to use one technique that can give us, in a label-free way, the chemical composition of the vesicles. And the technique that we use is called Raman spectroscopy. And this technique can be used in different ways. Let's say we can, for instance, capture our vesicles on a substrate, and or we can just have the vesicles in suspension, moving in Brownian motion, and then we can put our sample under our Raman microscope, which we can, and then we can actually detect a signal, and the signal that we detect is in fact what we call a Raman spectrum, is related to the chemical composition of the particles. So we hope that different particles will have a different chemical composition and will give us a different Raman fingerprint. And in this way, using these signals that we get by collecting the Raman signal, we could maybe label the different types of particles and detect the tumor-derived vesicles from other particles in plasma. We also did, because we already had the vesicles here captured, we also tried to do scanning electron microscopy and atomic force microscopy, just to look at the morphology and size, as this platform was already possible to image a different, moving it within different modalities. So I first will explain you a little bit about what is Raman spectroscopy. So when light interacts with the matter, for instance, with a particle, light can be absorbed, it can be transmitted, it can also be scattered. And this is the process that we are going to focus on, or that I was focusing on, the scattered light. So when, for instance, we have a laser that interacts with the sample, we will have a light that is scattered with the same energy, something that we call Rayleigh scattering or elastic scattering. But we will also have light that is of lower energy, that we call Stokes-Raman scattering. And these are the two types of signal that I will be detecting over, and they will come back in next slides. And let me just give you an example. So for instance, we have here a molecule. This molecule is, for instance, toluene. When we have a laser that interacts with the molecule, then we will have light scattered in different ways. The Raman light, we can collect it, and then we will see some sort of signal like this one, that what we call the Raman spectrum. And this Raman spectrum is specific to the toluene molecule, but other molecules will have a different Raman spectrum, so different peaks at different locations. And this is because molecules have exhibit different vibrational modes. So when light interacts with them, it interacts with the vibrational modes, and then the scatter light, the Raman light, depends on those vibrational modes. So that's where the Raman specificity lies on. So this is, in a way, a label-free, it's actually a label-free characterization technique, and that can give us information about the molecules present in the sample. For instance, we have a vesicle. The vesicle will have different molecules like lipids, proteins. So the Raman spectrum that we will see will be a combination of all of those Raman spectrum. But we hope that this combination will serve as a fingerprint to actually detect, for instance, the tumor-derived extracellular vesicles. Of course, Raman is a very low throughput technique, and this is challenging for the clinic, and I will come back to that later on. So the first thing that we did was to try to do, for instance, scanning electron microscopy and Raman spectroscopy on a sample, but first of cells, because cells are a lot easier to work with than vesicles. So we first wanted to test the method using cells. So for instance, here we have cells, and we can just have a fast scan just to look for regions of interest where we have the red blood cells. Then we use a microscope that actually a Raman microscope is embedded in the vacuum chamber of the scanning electron microscope. And then we can move to that region of interest. We can move our sample under the Raman objective. And there we can take a bright field image and to select the cells of interest or the particles of interest. And then we scan the sample and we obtain the Raman spectrum. So we obtain a single Raman spectrum per pixel. And then we can move back to the SEM and take a high resolution SEM image. And why we do the SEM after the Raman? Well, the thing is, like, when we have high resolution SEM imaging, we have to expose the sample a lot longer time to the SEM. And this can also cause electron beam induced deposition of amorphous carbon. And this can have some impact on the background that we get in the Raman signal. So that's why we do it after measuring the Raman. Then we can actually analyze the data that comes from the Raman. So all the Raman spectra, we will analyze it. And by using some machine learning techniques, for instance, we can use hierarchical cluster analysis. We can cluster the pixels that have similar chemical composition. So here you see cells, red blood cells, and the pixel with the same color corresponds to regions of similar chemical composition. And then you can look at the Raman spectra of these clusters here, and then start assigning which kind of biomolecules are present. And the next thing, you can also overlay the images. So we did that for instance, an unsorted sample of leukocytes, and we have here a same image. Here we don't distinguish which cell is which, but once we do the Raman imaging and the cluster analysis, we can label the different types of cells that we have in our samples, such as lymphocytes or neutrophilic granulocytes. And then we can look at the Raman spectrum, and interesting in this sample was that the presence of the nucleic acids, and because of course the lymphocytes have a lot of nucleic acids. And it was interesting to also look at these biomolecules and the difference between the two types of cells. So now we move towards vesicles, and this is quite challenging because cells can just sediment. You can also wash them easily. You can pellet them. But with vesicles, it is not that simple. They cannot sediment. You really have to capture them. And if you need to do washing, well, it's also not that straightforward. So what we did was to basically attach some antibodies to a substrate, and then these antibodies against specific antigens in the vesicles of interest. And then we can, once the vesicles are attached to the substrate, we can actually perform the Raman spectroscopy or SEM imaging or AFM. And the idea was to have some sort of platform that we can then use to be measured with different modalities. Not only to measure the same sample, but actually we wanted to measure the same vesicle, but with different modalities to get a lot more information from that particle. So we functionalize stainless steel, which is a substrate that is optically quiet, does not give so much background for Raman, and also is electrically conductive to take SEM images. Where we functionalize it with a linker, CDPA, that binds to an antibody. In this case, we use anti-EDCAM, that is targeting the EDCAM molecules in LNCAP-derived vesicles. And here we have some of the SEM AFM images. Here we cannot distinguish which type of particles are there. We just see morphology and sizes, but we don't really see what is their nature. But with Raman, we can get some sort of images. These are images are done after using a technique called principal component analysis. And what we see here is that there's a heat map, and the pixels with very high values, like this bright pixel here, can be described with this, let's say, Raman contribution, with this Raman spectrum in a way. And what we see here is a clear lipid composition of these particles. But there were other particles that clearly do not resemble like any biological nanoparticle. And they have also these particles that you see here. They could be described with this profile, which was showing that, in fact, this was debris from the microfluidic channels, the PDMS or alignment markers that we were using. And this was surprising to us, and it's of course not what we wanted. You don't want to have these particles or particles co-isolating with your vesicles of interest. But it was also nice to realize that by using more techniques, and in this case Raman, we were able to really discriminate between the particles of interest, in this case the biological and nanoparticles, which are our vesicles, from other non-biological particles in our sample. Now, this we moved from this capturing of vesicles in substrates. It is the process is long, and it's a little bit like it has to be optimized, and capturing it's always difficult because the capturing efficiency depends a lot on your linkers. So we decided to maybe try to measure the vesicles directly in suspension. And for this, we used a technique which is called optical trapping, or it can also be called laser tweezers. And the nice thing of this technique is that we can trap vesicles with the light. So how it works is that we can put a sample of vesicles and these vesicles will move in Brownian motion, and when they pass close to the laser and focal spot, they will feel a net force that will drag them toward the center of the focal spot. And this technique was developed by Arthur Ashkin. And what happens is that when you have a highly focused beam, so you can focus it with, for instance, a high numerical aperture objective, and also enough power, there is a transfer of momentum from the light to the particle. So basically, this will result in a net force that drags the particle towards the center of the focal spot. And this can be in the Z direction, but also it is in the XY direction. So we could use this technique to trap the particles, but at the same time, using the same laser, we can actually acquire the Raman scattering and also the Rayleigh scattering. And why we are acquiring the Rayleigh scattering? Well, the thing is, like, we wanted to know when a single particle enters the focal spot to measure actually single particles and see the moment when we have only single particles in our focal spot. So every time a particle enters the focal spot, the Rayleigh scattering is it has a rapid increase. A second particle enters and you see another rapid increase and so on. So if we integrate the signal and plot it over time, we will have some sort of a time trace, what we call a time trace, which is has a stepwise trend. And every step is a moment when a particle enters the focal spot. So we can see the moment when we have one particle in the focal spot, two particles, and so on. Now, simultaneously, we can also acquire the Raman spectrum of those particles. So here we see the Raman spectrum of a single particle up to 3 particles. And we know we can assign this Raman spectrum to the specific moments when we have only a single particle. And then we will know what is this fingerprint of single particles. We can also, for instance, integrate this specific Raman peak. We see that some vesicles have a clear unique peak. We can integrate it and then also plot it over time. And then also generate a time trace, Raman time trace. And if the Raman time trace overlaps with the Rayleigh time trace, then we are trapping a particle of interest. But if, for instance, we see that the Rayleigh time trace shows a step, but the Raman does not, then we see that we trap a particle, but it's not the one that we were looking for. By the way, Rayleigh scattering it has many names, elastic scattering, but it can also be related to, for instance, people might know it from the flow cytometry field, forward scattering, side scattering, or from MPA as well, the same type of elastic scattering. So we did this, we can do this actually over time. So basically at some point when we saturate the focal spot, we can close the shutter to the laser, and then the particle will be released. And then again, open the shutter, and then we collect more particles. And this can be done over time, so we can probe in this way our sample. So the first thing to do was to try to see if it works with a monodispersed sample that is well characterized, such as polystyrene beads. So we try polystyrene beads of 100 nanometers in this case, and we see here the time trace, the Rayleigh and Raman, and we see clear the steps every time a single particle enters. So if we have a zoom in, we see we can see the moment when we have only a single bead or two, and so on. And then we can get the Rayleigh scattering signal for each of the moments, each of the moments here. And if we want, for instance, we can also get the Raman spectrum of each individual particle. For instance, if we want to get the Raman spectrum, want to know the chemical composition of this particle, and that was trapped here in the third moment, then we can basically get the Raman spectrum of this one and subtract the contribution of the Raman spectrum of this moment, and then we can in that way get the fingerprint of single particles. Also, for instance, if our sample has a lot of soluble proteins, which can happen in plasma, the soluble proteins, they are very small to be trapped in our with our setup. So, but they will still generate some contribution to the Raman signal. We might see some signal. So that's what we did to solve that was to basically get the contribution of the baseline basically here, which is the contribution of all the particles, soluble proteins that were before a particle was trapped, and subtract that contribution for the next particle that comes. And in that way, we will only have the Raman spectrum from the single particle. So we tried this with different beads, different sizes of beads. In this case, it's an example of 125 nanometer polystyrene beads. We can here segment the signal, and then, for instance, we can, if we plot in this here the median value of each segment, we see clear levels that corresponds to the baseline here in black, and then in blue we see the first trapping event, and in green we see the second trapping event, and so on. So together with Edwin van der Pol, we try to calibrate our setup to, we are obtaining here the Rayleigh scattering, which is a backscattering intensity in arbitrary units. We wanted to calibrate it to get the scattering cross-section in absolute units. but also we wanted to really see if our data was in accordance with a me model. So in this plot, you see a me model. The theory are the solid lines, the black and red, for polystyrene and silica respectively. And then we also here feeded the data that we have here. So to basically show that, okay, we trap particles of different sizes, and we can relate perhaps the backscattering intensity with arbitrary units to absolute units. And this will come in handy for the vesicle data. So the next step was now to basically try our method for prostate cancer-derived vesicles. And we started with cell lines, in this case, PC3-derived vesicles. And we again collected the time traces. You see the clear steps. Here we see, for instance, a zoom in, and we see every time we trap a single particle, we have a step. And then we can get the Raman spectrum of each of these individual particles here. And then you can already start seeing protein contributions, lipid contributions, some specific amino acids. So you can test the heterogeneity of your particles. And well, if we do just an average of all the particles that we trap, we can have maybe a fingerprint for that specific population, and start looking at lipid to protein ratios, for instance. So we then wanted to do this for different type of extracellular vesicles and also other particles in plasma. So here we see the mean Raman spectrum, so the mean Raman profile of different type of vesicles, in this case red blood cell derived vesicles, two types of prostate cancer derived vesicles, which are PC3 and LNCAP, and also particles that are present in plasma, and also particles that are present in platelet concentrates. And this is just the average of the Raman spectrum of all the particles that we trap during one of these experiments. So some of the interesting things that we can already see is, for instance, that the protein to lipid ratio is larger for the vesicles, as we can see from this, for instance, this peak and this peak, compared to the particles that are present in plasma, which we will see later on that the majority are actually lipoprotein particles that we are trapping. We see clear phenylalanine peaks for the vesicles here, with it's not really detectable for the lipoprotein, for instance. But okay, we also need not only a single particle detection technique, but also we need a single particle analysis method. So for this, we use principal component analysis, which is a method that can help us to see the largest variations in our data set. And we can distinguish maybe what are the specific features that make the vesicles different from each other. So here in this scatter plot, you see that each dot here represents a single particle. So here we have that red blood cell vesicles clustered together, separate from the prostate cancer-derived vesicles, which were here clustering together. And these ones cluster separate from the particles in the platelet concentrates, which in fact cluster together with the particles in the plasma, which it makes sense when we later on, because we know that the majority of the particles that we were trapping here were actually lipoprotein particles, and they were all clustering together. So this was already interesting to see that, at least from the vesicles that we use from cell lines, we could differentiate them from other vesicles or other lipoproteins present in plasma. So if we look a little bit more in detail at the PCA analysis, so this is a scourge plot, and then here on the right we have something that we call principal component loadings. And basically what this says is that if the particles have, for instance, high values for this y-axis, which is the principal component 2, they will be they can be mainly described by, for instance, principal component number 2. And this principal component shows a lot of a very clear protein contribution. So in fact, particles with high numbers for PC2 here will have a large protein contribution. So we see here that, for instance, red blood cell derived vesicles and most of the vesicles from the prostate cancer were also having a large protein contribution, while for the case of the lipoproteins, that was not that much. Now, we wanted to see actually the origin of these particles that we took from the plasma and the platelet concentrates. So we characterized commercially available lipoproteins. We thought of first using only chylopicrons and very low density lipoproteins because they overlap in size and density with the vesicles, and also these will be the particles that can be trappable with our setup. So that way we started by characterizing this, and you can clearly see that the fingerprint is very much alike like the one from the particles present in the plasma and platelet concentrates. So in fact, the particles that we were trapping there were mainly lipoprotein particles. Now, the next thing we wanted to see, we could predict the sizes of the vesicles that we were trapping based on the Rayleigh scattering, and using also the me model. So together with Edwin van der Pol, what we did is to use again the same plots that we had before to basically calibrate our setup. And then we basically plotted a line here in green that is the predicted scattering for the vesicles, varying, for instance, the refractive index of the core, and with a specific shell thickness, and also a specific refractive index for the shell. And what we see is that it is not so easy to predict the sizes based on the backscattering intensity that we get. So here we get, for instance, different values of the backscattering intensity for the different particles. But if, for instance, you have a particle that has maybe 10 to the 5 as value here, then you see that if we look at this axis, then basically the particle 10 to the 5 can intersect with at many locations with our predicted model. So we don't have a unique answer for a size based on a single value of the backscattering, and that is a pity. There is very sharp. These lobes are very prominent. So, but at least we can have a size range of the particles that we are trapping. And what was very important for us was to see that we were really trapping soft micrometer particles, and that the model could help with. By looking at this gray line, is basically the distribution of the Rayleigh scattering values of the vesicles. And if we look, for instance, at the median values, we see that most of the particles that we are trapping are soft micrometer particles. And that was for us important to know. And also using this kind of model, we can, for instance, give a size estimation for the detection limit. In our case, again, the detection limit is just has a size range, a broad size range. So that is not ideal. We would like to have a single value for the detection limit of our system. But based on the polystyrene and silica measurement, we think that we are measuring between 100 to 200 nanometer particles. So I think it is important what's important is still to see the limitations of our setup in terms of the size of the particles that we are trapping. The next step was basically to go and characterize and detect particles in the plasma of cancer patients. In this case, we did a small pilot study on metastatic castration-resistant prostate cancer patients, and compared those particles that we trapped from their plasma with healthy donor plasma. So we again isolated the plasma, and then we did the optical trapping and Raman spectroscopy. And here I will show you some average Raman profile that we see from first the healthy donors. We see that most of the particles that we are trapping are actually lipoprotein particles, as we had expected. Still, the fingerprints these Raman fingerprints are not. There are some variation. There are some variation between the healthy donors, and that is already interesting. Because that can depend on many things, but it will be nice to actually profile these particles and dig more in detail to see what kind of lipoproteins are there, or the heterogeneity of these lipoprotein particles, for instance. And now we can also compare them with a different type of lipoprotein particles. We in this time we added also low density lipoproteins and also the high density lipoproteins. And then we can already see here some clear distinctions between in the Raman profile between the different lipoproteins. For instance, the triglycerides are very prominent for the chylomicrons on the VLDL, while cholesterol esters are a little more prominent for the LDL and HDL. And now I will show you this mean Raman spectra of the patients. And here's actually very interesting because we were expecting maybe to have a Raman profile that is just like a lipoprotein profile. And for some patients was the case, patient 1 and 5, but there were some patients, patient 2, 3, and 4, which showed a fingerprint that was clearly different from the rest of the patients and also healthy donors. So we did principal component analysis, and we saw then that some of the patients were really clustering far from the healthy donors, especially patient 4. And we wanted to see what was making patient 4 so different from the other patients or the other healthy donors. So we looked at the principal component loadings, and in this case, in patient 4, we saw that had very high values for principal component number 2. And if we look at this principal component loading, then we see that there are many peaks here that are related to nucleic acids. So in fact, patient 4 had the particles that we were trapping from patient 4 had a large contribution of nucleic acids, which was very surprising for us, especially because we were using very small volumes of plasma and we didn't do any depletion of lipoproteins. So now we did the principal component analysis, but now taking into account many more particles, like the lipoprotein particles, but also the prostate cancer derived vesicles, which are actually a model for this type of cancer that we were analyzing. And to our surprise, or maybe not, the tumor-derived vesicles from the cell lines, they were not overlapping at all with any of the patients. And what also later we did was to do hierarchical cluster analysis. This is a technique, like I mentioned, that can help us to cluster particles that have similar chemical compositions. And I use the same representation as this one, but in this case, the colors are refers to particles with similar chemical composition. So we see, for instance, that the patient 4 is clustering on its own cluster, separate from the rest. We see interesting things, for instance, that patient 5 is clustering with HDL and LDL particles. When we did a lipid analysis of these particles from patient 5, we saw that the total cholesterol and the LDL particles were the highest from all the samples that we measured. So that was nice and reassuring that we were able to also detect that, but using, in this case, very small volumes. And then the VLDL and chylomicrons were mainly clustering with the healthy donors, as we would expect. Still, there were other clusters present in the healthy donors. We still have a lot to learn from this. We are still working on this because I think that the Raman profile or the particle profile in these samples is very heterogeneous and even already in the healthy donors. So now, if we look, for instance, at the specific Raman fingerprint of different clusters, we see that, again, for cluster 8, which corresponds to patient 4, we see a lot of contribution of nucleic acids. And we actually analyzed this also for circulating tumor DNA, and this patient 4 had the largest contribution of circulating tumor DNA in plasma, and also, interesting, the highest PSA value, prostate-specific antigen. So it was already very interesting, but also because the profile that we see is also associated with lipids. And we actually trap particles, and I don't think with our setup we can trap just the circulating tumor DNA with the histones because it's too small to be trapped. But we do trap particles, and we see lipids and nucleic acids. So it might be that the particles that we are trapping are associated, are lipid particles, either lipoproteins or vesicles, associated to nucleic acids. And this is something that we have to prove further, but it's still interesting to notice. So now to conclude, and I will check out, didn't check out the time, but okay, we are on time. Well, this method of optical trapping and Raman spectroscopy, it's nice for the detection and chemical characterization. I would say the global chemical characterization of biological nanoparticles at the single particle level and as a label-free technique. It can also, it works for discriminating different type of vesicles or lipoprotein particles, and maybe also for purity assessment of our vesicle samples. One of the disadvantages is the throughput. The throughput is very slow, especially compared to flow cytometry, which is very used, for instance, in the clinic. But still, I think it's nice to see the general profile of particles, of sub-micrometer particles in plasma of patients, either with this optical trapping Raman spectroscopy techniques or many others, because also the particle profile might be affected in different diseases. And now I would like to just raise some discussion points, and this is related to sample preparation. So we were aiming at detecting the tumor-derived vesicles in plasma. And this plot, we see the concentration against the size and the density of particles in the plasma. This is taken from values from the literature. So we here see, for instance, vesicles that are derived from the endothelial cells or white blood cells, red blood cells. And this is, so we have these vesicles with maybe a concentration of up to 10 to the 11 particles per milliliter. Now, if we look at the tumor-derived vesicles, that's already a lot lower, maybe with the small tumor-derived vesicles having a concentration of up to 10 to the 4 particles per milliliter, and the large ones even less. Well, we have here some platelets, but the main problem are actually the lipoprotein particles, which if we look at the concentration, they are maybe 10 to the 9 or 10 to the 10 times more concentrated than the rare populations of tumor-derived vesicles. So it is very challenging to detect these vesicles in the pool of, in the abundant presence of these lipoprotein particles. And there are very nice techniques that we can play around. For instance, size exclusion chromatography could help us to remove the small lipoprotein particles. Then if we look at the density, we see that these large lipoprotein particles are very different from vesicles. So we could use, for instance, density gradients to deplete the large lipoprotein particles, and then in that way try to isolate only the vesicles. But one thing that I think is important to be aware of is that every time we deplete our plasma samples from lipoprotein particles, we are also losing vesicles in the process, and maybe we're also losing the vesicles of interest, such as the rare population of these vesicles. So it is clearly needed a specific enrichment of tumor-derived vesicles in plasma. So not only depletion of the lipoprotein particles, but also a specific enrichment of these tumor-derived vesicles. And another thing that we saw was we wanted to see the detection limit of our setup in terms of frequency of tumor-derived vesicles that we could detect among the pool of lipoproteins. So we were mixing prostate cancer-derived vesicles with lipoproteins, actually in plasma. And when we look at the scatter plot, in all the experiments that we did, we always saw that part of those prostate cancer-derived, actually part of that mixed sample was clustering with the prostate cancer-derived vesicles, and the other part with the plasma suspected. But there were always particles falling in between the clusters. And when analyzing these clusters in between, we saw that the Raman fingerprint, which you can see it here on top, was showing a contribution of both the spectrum, the lipoproteins or the proteins in the plasma, and the vesicles from PC3 vesicles. So this could maybe suggest that when the PC3 vesicles are in the presence of many other soluble proteins, they can associate to them and form these protein coronas, or maybe they can associate to lipoprotein particles. And in the literature, some people have already reported some of these cases. But if this is really happening, it is very important to be aware of, because this will block potential binding sites, for instance, of the tumor-derived vesicles for specific enrichment. So think it's something to be aware of. And then the last point, my last slide is basically, is there a preferred way of performing single particle characterization? Is it capture or in suspension? I think ideally we would like to have maybe just the plasma and measure it directly and just get the information from the particles there. But I think also it would depend a lot on the techniques that we use. And one of the nice things to do would be to have sort of a platform that we can not only measure the same samples with different modalities, with different techniques, but actually measure the same vesicles. So with this, I would like to thank a lot my mentors, Leon and Case. I've been working and learning a lot from them, and I keep learning a lot from them. Also my collaborators from the AMC, Edwin, Ring, Ton, Chi. Also from the AMC, from the Medical Cell Biophysics group, Professor Arman, I would like to thank a lot. And of course, all my colleagues at the Cancer ID Consortium and the funding agents. And I would also like to thank you very much for your attention.
All right, thank you, Agustin, for the clear explanation. I think this was an illuminating talk. So there were already a couple of questions and you can keep sending them in. So the first one was from Juan Alfonso. Do you want to unmute yourself?
Yes, can you hear me clearly?
Yes.
Okay, thank you very much. It was a very nice presentation, Agustin.
Thank you very much.
My question regards to how is it that you can discriminate between extracellular vesicles and other non-EV particles? You sort of answered my question in the last part of the presentation, but what I trying to ask is, with the sole Raman scattering is enough to separate EVs from non-EVs, or do you have to do additional characterization to determine?
Yeah, thank you very much for your question. It's a very interesting point actually, because when we characterize where a lot of biological nanoparticles, and then we can get a Raman fingerprint, and this already gives us an idea of what a Raman fingerprint for these biological nanoparticles looks like. But it is true that if we have other non-biological nanoparticles or other type of particles, we will just see it as a different spectrum. We can start looking into the details of the spectrum and look at the specific vibrational modes and assign some molecules to this spectrum to try to guess what kind of material, what kind of particles are we treating. But I think it is also important to, for instance, characterize control samples. Like if we run a size exclusion column and then we just run it with PBS, we get it, the PBS out, and then maybe there are particles, maybe not, from the material. And then it will be nice to characterize all of these controls, and then we have a fingerprint for the different controls, and that can help us even more to differentiate between our vesicles of interest and other types of particles.
I was going to suggest, just, I don't know if you did it, but you could do, obviously it doesn't represent exactly a tumor, but you could do cell culturing with serum-free media, and maybe that would help you to have like a control sample to characterize EVs. Yeah,
that's a good point. In fact, the vesicles that we measured were all cultured with the serum-free medium, and that was very important, of course, because we also characterize FBS particles, and main composition there is actually lipoproteins. There are many lipoproteins in the serum. So it's a very good point, and we did mixtures of it. We also did vesicles characterized without the serum to be sure that what we are measuring is the vesicles. Yeah.
Okay.
Yeah.
Thank you very much.
Okay. The next question is from Lilia, but I think has that been answered already?
Not really. Yeah, thank you for the talk. It's great. It's just a very naive and simple question that, for instance, if you got the agglomerate of the small particles together and you have in the same solution the large particle in the same size of that agglomerate, so the membrane structure is the same, if there is a way to distinguish between them?
Yeah, that is a really good point. Thank you for the question. Yeah, so in fact, if the particles are already clustered, we will trap them as a single particle. We will see the Rayleigh scattering as a single particle. But the thing is, like, Rayleigh scattering also depends a lot on the geometry. So what we have seen is that when we trap a spherical particle, we have a Rayleigh profile that is quite, let's say, linear in a way. But when we trap a second part, basically two particles together, or that were in a cluster, or three, it's not anymore a spherical particle. So the Rayleigh scattering profile changes, and this is visible in the Rayleigh scattering. And we are actually also working on this to try to detect clusters from the Rayleigh signal, from analyzing these profiles, because of this interesting phenomenon that we see, that the geometry affects the Rayleigh signal that we get. Now, the other thing is that in the Raman, we have the same composition. For the Raman, it is different to distinguish if we have a cluster of particles with exactly the same composition as a big particle that is the same size, because Raman is directly dependent on the amount of molecules in our sample. So if you have the same molecules and the same numbers, we'll see it the same. But Rayleigh can depend on this geometry property. So in this way, we could be able to eventually distinguish them. Yeah.
Okay, thank you.
Thank you.
All right, the next, Norman is wondering how you can relate the spectra to cells of origin. So
Yeah.
please, Norman, go ahead.
I think in his answer to the first question, he addressed my question. So thank you.
Yeah, all right. Yeah, no problem.
So the next one is from Celine. Are you there?
I think Celine has dropped off, but
Okay.
she's asking about the concentration of EVs. What was the concentration of EVs from different cell types that were used to generate the spectra of slide 23? So
I have it here, no problem. Slide 23.
I guess just in general,
Yeah,
you know, how,
right. Yeah. Okay.
How many EVs do you
exactly.
really need?
Yeah. So well, the one thing is the concentration in the sample, and the other thing is the particles that we actually probe, because we cannot probe all of the particles because of the low throughput of the Raman, but we are probing around 100 to 200 particles per cell line, per sample, let's say. We can do duplicates, or we can just do longer time traces to probe more. So at the end, I think the last analysis that we had in total, there were like maybe around 2,000 to 3,000 particles involved, single particles. Yeah.
Okay, so then there was an excellent comment from Madhu. Are you there, Madhu?
Oh, yeah. Yeah, I'm here. Sorry, I'm muted. Excellent presentation, sir. I have a small question regarding, like, so as we know, like, a cell produces different populations of EVs. Maybe like also the lipid composition of these different populations are different. And is it comparable to the cell they are derived from, the lipid fingerprint to the cells from where they are coming from? And maybe it's comparable, or did you ever looked into that profile?
Yes, that's a very good point. We actually have a look at that, because we also did the Raman imaging of the cell lines, of the cells where we produce the vesicles from. The lipid composition is very similar, in fact, surprisingly similar. Of course, for the cells, we use confocal Raman microscopy, so it depends on at which location of the cell we are, the Raman spectrum that we will get. So if we are at the nucleus, we will get a large contribution of nucleic acid, but if we are at the membrane side, then we will have a lipid composition that can resemble the vesicles. And this was very similar. So this is actually a very good point, and something that is interesting to keep doing. Indeed. Thank you. Yeah.
Thank you very much.
Great. Next up is Randy, who is using a similar technique to analyze his EVs.
Hey, August.
Randy.
Yeah, good
Nice
to see you.
Yeah.
Yeah. You know, I probably have 10,000 questions, and we always have really nice discussions on this stuff, but I'll limit it just to a more general one.
Great.
So I had 2 points. I'll make them really quick. so we're typically using much higher power objectives like 60x, 100x, and we almost never see a second particle come in. So I'm wondering if that's something you think is happening, or second particles come in, but they bump the first one out basically. And you guys see 2, 3, probably even more trapping events. So I'm wondering just about the optics, your laser power objective, if you think that's the case.
Yeah.
And then yeah, you can go with that one first.
Okay. So yeah. So so basically we are using laser powers of 70 milliwatts. In our case, we use a numerical aperture objective of 0.95, and we are using 40x objective. The thing is that it also depends on the size of the particles that we trap. So when we go to, for instance, the 400 nanometer bits that we test in the beginning, or 800, then yeah, we'd only see one single particle being trapped. But the small ones, we really see like a second, third, and also for the vesicles, the small ones we see this couple of particles until we saturate the focal spot. Yeah.
Yeah, cool. That's pretty interesting. I guess my other point is, in our recent work, we started to, when we dig into these complex spectrum, I think some other people are already starting to to ask questions about the subtleties of how you can tell these things apart. We found that the sugars, we started seeing lots of molecules related to sugars, and we think coming from glycocalyx, we can chop them off with different enzymes, and we see these signals dropping out. And especially with our cancer patients, similar to what you showed at the end, we saw some influence. I was wondering if you saw any of those chemical functional groups or have looked in that direction.
That's actually really interesting. We haven't seen it. That's also ongoing progress. We haven't seen the sugars. We now focus on this interesting finding of the nucleic acid, but I definitely will also have a look at your very interesting findings of the sugar. Yeah, sounds really, really interesting.
Cool. I just want to say too, one thing, I mean, the Raman stuff is all cool, but the first part of the talk, whenever, you know, we're so focused on these single particle methods, but your single particle methods are doing, you know, SEM, AFM, and Raman, but on the same single particle. That's really different from a lot of the other single particles. So I just find that stuff really cool. So good job.
Yeah, thank you. Thank you. That's what we wanted, or at least we think that you can get a lot of information from the different modalities. It is very challenging, and we still keep optimizing on that sense, because just the transport of the sample, you know, what are the storage conditions, how much do you measure it with one setup to have a vesicle that is still viable for a next setup. So there are many challenges there. Also, for instance, scanning electron microscopy, if you measure in vacuum chamber, we have to dry the sample. So we have tried different drying techniques, from chemical drying to critical film drying. So it is challenging, but I think it's nice to have all of this information of the same particle.
All right, thanks, Agustin.
Thank you. Thank you, Randy. Nice seeing you.
Okay, so I think it's time for the last question. Estefania, are you there?
Yes, I'm here. Can you hear me?
Yes.
So great talk, Agustin. I was just wondering if you tried to validate the model with measuring particles smaller than 100 nanometer in size, and also if you tried to measure liposomes, for example, or even liposomes modified with the specific proteins, and then if you were able to identify these proteins back in the spectra.
Thank you very much for your very nice question, Estefania. Yeah, that's a very good point. So what we only try to do is using hollow silica bits, and kind of resemble, because the hollow silica bits, they also are filled with the same medium that you put them in, for instance, PBS. So in a way, they are a good model for the vesicles. We tried this one. We were able to trap them, but we didn't go further with liposomes. I think it's very interesting still to try liposomes and other particles. Yeah.
Great, thank you.
Yeah, thank you.
All right, it's time to wrap up. Agustin, thank you. Thanks a lot for your presentation. Everyone who called in today, thanks for your attention, and see you next week on the EV Journal Club.